All right, good morning, good morning. Get the camera in place. So today, we're going to talk about the hematopoietic system, which, for those of you who haven't worked out yet, and I think we've already talked about it, the hematopoietic system is the stem cell progeny for your blood. Stem cell progeny for your blood. So we're going to go through some of the history, how you refine it, the therapeutic uses of hematopoietic stem cells, and just kind of get a feeling for the space, the role. We're going to get a feeling for the role of hematopoietic stem cells as well. So, we should all be familiar with blood progeny, but this is a HSC, hematopoietic stem cell. And the hematopoietic stem cell gives rise to red blood cells, white blood cells, and platelets. I think that's how you spell platelets. Right, now there are a whole bunch of intermediate steps here which we're going to go through in a little bit of detail, but we're not going to look too closely at the progeny. Okay? We're going to look at the history, how it was discovered, the function, and the therapeutic use. Okay, so let's get on to the history. So history is the heading. Right? You can't see that? That's great. History. So, how did we kind of know that hematopoietic stem cells existed? Well, we already discussed a couple of times the radiation-based experiments in stem cell biology, but irradiation was kind of the first clue. So what did we do? Well, we take a mouse, we irradiate the whole body, the mouse eventually dies, right? Or we take a mouse and we shield the spleen or bone marrow sample, bone marrow part of bone, from the irradiation, and the mouse lives, right? Very simple. Very simple. And so, then we move on to the work of Till and McCulloch, which we've discussed many times, but it doesn't hurt to reinforce. Right? And as we know, what they did was irradiate one mouse, and they take a bone marrow sample from another mouse, and they see whether or not the irradiated mouse survives and indeed has a blood progeny. And then from that we can draw conclusions about where the blood progeny lies, and indeed we find it in hematopoietic stem cells. So now we're going to look at some of the assays. We looked at the history, now we're going to look very quickly at the assays that we use to determine stemminess and indeed whether a cell is a stem cell. So I have three assays. Colony forming unit. Right, I think it's the extended colony forming unit. Colony forming unit with differentiation. And we also have the in vivo approach. Right, cool. So the colony forming unit assay is really straightforward. We do pretty much the same thing as Till and McCulloch. It's fast, it's 8 to 12 days. So this one, oh, I wish I had a working pen. Oh, that one's nice. So it's 8 to 12 days, right? You can see that. And what we want to do is see whether injecting an irradiated mouse with new hematopoietic stem cells or bone marrow transplant will indeed form colonies on the spleen, right? And this is cool and all because it shows that we can get some progression from those cells to regenerate the blood system, but it's not exclusive. It's not exclusive to stem cells. You can actually work with progenitor cells as well because you put them in and there's not enough time for the progenitor cells to all die off before the stem cells have to take hold. So it's possible under this regimen that the stem cells are not actually doing the regeneration, it's the progenitor cells instead. Could be progenitor cells. Right? Cool. Could be progenitor cells. Right. Now the colony forming unit extended version is when we do the colony forming unit experiment multiple times. I'm going to pause. Are we in focus? Right. So in this one, and I needed to look it up in my notes, but in this approach we need to know that in fact the cells that we've injected from the donor are different to the host. So we have our mouse that's been irradiated and it has a slightly different type of CD34 marker to the host or to the donor, so that we can tell whether the colonies that we're forming on the spleen are indeed from the donated cells. And indeed, if they're from a specific donated cell, we can see if it's from one cell. And we've been through all of these kind of assays over time. Okay, so can differentiate. Probably differentiate is a bad word, but tell apart. Can tell apart donor host cells, right? And that's important because it lets us know whether indeed the donor cells are actually causing the blood progeny, and in fact they're not causing some cells that are already in the mouse to start to do their job again by some kind of signaling mechanism. And that's the alternative hypothesis, right? So we'll just put that down. Not signaling internal cells to work. To proliferate, to work, to do their function again. Right? And then the in vivo approach is the one that we use for humans. Right? And that's cell culture. Right? That's cell culture. And so the important thing of this one is, so it's 5 to 7 weeks. 5 to 7 weeks. 5 to 7 weeks. And in that duration, all the progenitor cells die. We've talked about this before, I remember. But 5 to 7 weeks, progenitor cells die. Therefore, we must have stem cells. Because if the progenitor cells have died, we have to have stem cells, right? There's no way that there's nothing else than they can be. So there's 3 assays that we're talking about with hematopoietic stem cells. So we've got the short one, which takes 8 to 12 days, and that allows us to see stemminess, somewhat stemmy. Then we've got the extended one, which with some differentiation, so the ability to tell apart the donor cells and the accepting body cells, the host cells. And that's important because it allows us to tell whether the colonies come from an individual, and also it allows us to know that the proliferation and the blood progeny is indeed coming from the cells that we inject and is not being elicited by the cells that we inject from the cells that are already in the mouse. right? So that's important, right? So the cells that we're injecting are not causing signaling, they're not causing signaling for the cells that are already there to work. They are in fact themselves doing the work. And finally, when we do it in vivo, we wait 5 to 7 weeks, because after 4 weeks, all the progenitor cells die. They only last 4 weeks in culture, which is why this one, which only lasts 8 to 12 days, can be caused by progenitor cells, right? And then this is the one that's mainly used in human experiments. Cool. Right. So, now that we've looked at the assays to determine whether something is STEMmy, let's look at how do we isolate a stem cell? How do we take a sample of tissue and we know that we have stem cells, okay? How do we isolate stem cells? How to... Right, and the first thing we're going to learn about is facts and these are heaps important. Facts and max. Facts and max, heaps important, really important. We need to remember these ones. So facts and max. So fluorescent activated cell sorting and magnetic activated cell sorting are two really important cell sorting, purification and enrichment approaches. Purification and enrichment. So you take a sample of cells and you go, I want cells which have this antigen, and I've attached our marker to that antigen, or so they have this antibody and we attach this antigen, and that allows us to pull out the cells that have that marker on them, right? So all it is attaching an antigen marker and being able to differentiate cells, or being able to tell apart cells based on their adhering to that marker. And you can think of them as, well, literally magnets is one of them, right? But facts, I suppose, you know, they're like the cyclist reflecty lights. And then if you have a cyclist that has two different kinds of reflecty lights, that's a special kind of cyclist. We'll get into it. Okay, so how does it work? Well, we have a tube. And like I said, our cells have a little magnetic antigen made with iron that we attach to it. And they all come through, and these ones get attached to the wall, and then we filter out these boys, take them away, and then we turn off the magnet and get the ones that we want. Magnetic cell sorting, magnetic activated cell sorting. So this is good for enrichment, but not for purification. And the downside is it's not that pure and you only have one marker, right? Max, not pure, one marker, right? But it does have the advantage of being cheap, and it does have the advantage of being high throughput. Right. And max is typically used in clinical applications. Cool? So that's magnetic activated cell sorting. So you take a cell, you put a antibody attached to it, which has an iron particle, an iron molecule, and that allows us then to magnetically sort out the cells. But we don't sort them purely, we just enrich. Okay, and then facts is we have a tube, right? And our little cell might have a green marker and a red marker, and we shine a laser on it, right? And the laser comes off into a receiver. Look at me, I'm a receiver. Very exciting, right? And so we individually read the signals from individual cells, right? We're trying to laser on individual cells, and we get a reading off individual cells, and it tells us this cell has this marker and or this marker and not that marker, right? And so if you have a specific cell type that has several markers, you can use several different colored markers with this. And I can't remember the word for the markers. Sorry. And so that's FACS. So FACS is slow, expensive, more pure, very pure, multiple markers. So you get why there's multiple markers, right? Because there's more than one color, but there's only one ion. There's only one magnet particle that we can do with that, okay? Right, there's multiple markers. It's slow, expensive, it's highly pure, and it's used for research. Because in research we need to have like actually the specific cells. Like we need to have actually definitely stem cells so that we can do research on the stem cells so that the results we get we know are coming from stem cells, as opposed to when you inject them into a patient and you're like, well, I only need it to do the thing that I needed to do. I only needed to perform the therapy. So as long as there are cells in there that do the right thing, that's cool. What should I add? No, that's next. Cool. So let's do a little bit more detail on the isolation of mouse and human isolation. Mouses, right. So this is a FACS plot that I'm drawing right now. And so we're going to have C kit, thigh 1, C kit and thigh 1. And then after that, you're going to have CD34. And what's the other one? Lin negative. And I've actually drawn this horribly wrong. Okay, so I've set up our FACS plot, right? And we're going to look at the different factors, the different markers, in terms of whether or not mouse hematopoietic stem cells are positive in these different markers. So, are they positive in SCAR1? Yes, they are. Are they positive in c-KIT? Yes, they are. Remember that. So that means that we want to be looking in this area of the plot. This area here. And so it turns out, whatever, it's a wiggly kind of circle thing. But what that means is we take these cells here. Take those there cells, because they pass our test for whether or not they're positive in c-KIT and they're positive in SCAR1. Cool? Then we take those cells and we stick them in here and we test them for Lin and SCAR and Thigh1. Lin and Thigh1. Now, are they positive in Lin markers? I'm going to talk about Lin markers in a second. But they're negative in Lin markers, right? So they're going to be down here and then we're going to be positive in Thigh1. And so we get something like this, and then we take all these cells. And that's like the stemmiest cells that we can get. The stemmiest cells that we have available to us. But that's going to give us about 1 in 3. So out of these cells, 1 in 3 are stem cells. Which is surprisingly low, I thought. But that's the level of purity that we get. It is what it is. So CD34. So, yep. And that's how we purify mouse stem cells. Now, by contrast, how do we purify human stem cells? Remembering that many of these markers don't necessarily play a direct role in stemminess. So in humans, right, we do size, and that's not really important because it doesn't actually differentiate them at all. Okay, so we do size of the cells, we do lineage negative, we do Thigh1 positive, that says Thigh1, and we do CD34 positive? negative. We do CD34 negative. CD34 positive in humans. CD34 positive in humans. Okay, so we have this guy, right, and we take these ones, stick them over here, and we get something that looks like this, and we take these guys here. We actually take the, but it's only that. And then that gives us our more purified human hematopoietic stem cells. Okay, so now that we've learned how to isolate human and mouse hematopoietic stem cells, we're going to talk about what makes a stem cell in terms of what's inside it, in terms of its soul. And so what that means is stem cells live in the same environment as all the other cells. They live in the same place, they get the same signals. So how do they know that they are stem cells, right? Well, obviously there's genes. There are transcription factors that activate genes that tell them I am a stem cell, right? So what are the critical genes for being a stem cell? It is SOX8, SOX18, and those are critical for maintaining stem cell state. We also have NFI beta, MYC, and IKZF. Right? And those are good for differentiation. Okay? Now you don't need to memorize those, but it might be nice to memorize them if you want to. But the really important group, and I want you to say this with me at your computer, the really important group is the BMI polycomb group 1. BMI 1 polycomb group, right? Write it in big F off letters on your text, on your notebook, okay? BMI polycomb group. BMI 1 polycomb group. This is the thing. This is what tells, and this is something that I found fascinating, right? Because cells in large part are very much regulated by their environment, and we have the ability to cause cells to become stem cells, right? But there's this really powerful signaling system that tells a stem cell that it is a stem cell and keeps it as a stem cell. But again, it lives in the same environment, gets the same signals. So how does it know it's a stem cell? BMI polycomb group 1. Write it down. Real important. BMI polycomb group 1. BMI polycomb group 1. Big deal. Big deal, big deal, big deal. So, what happens when we overexpress polycomb group 1? Well, you get superstems. So this is BMI1 going up. Get real STEMmy behavior. And then if you have BMI polycomb group 1, BMI1 down, right? You get the loss of clonal potential. Loss of clonal potential. Right? And eventually death in mice. Mice die if you turn off the BMI polycomb group 1 line because it stops the stem cells stem celling. Right? So that's a conformational experiment, right? Because when we turn it off, we find that the cells stop being stem cells. And indeed, that means that this, at least in part, is responsible for it being a stem cell. Cool. All right, we're most of the way there. So very quickly, we'll talk about therapeutics. So it'd be nice if we could use these hematopoietic stem cells to treat leukemia. Obviously, leukemia is cancer of the blood. And so I'm not sure if I've talked about this before, but we're going to talk about it. So in the blood cell lineage, right? So this is a long-term hematopoietic stem cell, this is a short-term, long-term, short-term, HSC or BSP, blood cell progenitor. But if you have cancer of a given cell, the one that comes after it in the blood lineage, you have too many of, okay? So if you have lymphocytic progenitors, if you have cancer of the lymphocytic progenitors, you have too many lymphocytes. Making sense. If you have cancer of this guy, blood cell progenitor, you get too many lymphocytes or you get too many myocytes. Cool! Or lymphocyte progenitors or myocyte progenitors. Sorry. So then let's talk about therapeutics. The thing about therapeutics with these cells is they're hard to culture. It's really difficult to culture hematopoietic stem cells in a dish because they're non-adherent and they like to float around and they die. So you in theory could take some cells from your sibling or identical twin or sibling and we would try and culture them and re-inject them into you, but they'd probably die, so that's not the best way to go. How do we get them? Cord blood. So, little umbilical cord blood, blood of donors, right? And so how we do that is we give the donor a drug that causes the hematopoietic stem cells to go into the blood, and then they roam around and we take out some blood and we refine the hematopoietic stem cells and use them for therapeutic purposes. And then finally, and this is kind of the old-fashioned way of doing it, is a bone marrow sample. Bone marrow sample. And that's quite a big commitment. Like, it's a big deal to get a bone marrow transplant or to take a bone marrow sample from someone. Like, it's a big FO needle and you shove it into someone's hip, and it can be quite debilitating and extremely painful. So it's not something that you just do. It's not just like donating blood. It's a big deal. So that's the thing. And yeah, of course, we'd like to use these to treat cancer. Cancer, if it's of that lineage, as we said before, you want to knock out that lineage, kill the stem cells, and then put in new stem cells of the lineage of the correct variety, right? So you can have autologous transplants, which is from me to me, from yourself to yourself, and allogenic. So autogenic and allogenic? Yes, autogenic and allogenic, right? And so if I take a donation from myself and I had leukemia before the donation, we couldn't filter out all of the cancerous stem cells, all you need to do is inject one cancerous stem cell back in and you get blood cancer again, even though I've had all this chemotherapy. If we take it from another person, so autogenic and allogenic, right? So autogenic, what I said before about getting cancer, okay? Not great. And in the allogenic, so a way to remember autogenic and allogenic is I drive an automatic car and I rub aloe vera on my girlfriend. So allogenic transplants from other people have a 60 to 80% chance of getting graft versus host disease, right? So 60 to 80% GVH disease. Now that's big, that's a lot. That's a hell of a lot, right? So there's a 60 to 80% chance that if I give you my hematopoietic stem cells and we match in terms of our immune blood type, that my hematopoietic stem cells are going to attack you anyway. And you're going to need to spend the rest of your life on immune suppressants because you got these hematopoietic stem cells, which is not great. Not great. And so yeah, we use that to treat cancer, we use it to test drugs, we use it to see how your disease is going to progress. And that is our introduction to hematopoietic stem cells. Thanks, guys.